to see it's actually really nice to see so many of you here. And this is our first ever seminar series for the ESEB Special Topic Network on the joint evolution of mating systems and dispersal. And so I will briefly introduce our first speaker, Greta Bocedi. So Greta did her PhD and postdoc at the University of Aberdeen, and she's now a Royal Society University Research Fellow there. And her research focuses on understanding how species adapt and respond to environmental changes by using ecological and evolutionary modeling to develop new ecoevolutionary theory and theory-driven applications. And one of her key interests is the coevolution of dispersal and mating systems, which inspired her to apply for the grant of ESEB Special Topic Network. Her goal is bring together all researchers from different fields studying dispersal and mating systems to explore how those two traits co-evolve. And that is actually the reason why we are here and also brought us all here today. And now I will hand over the stage to Greta, and she will introduce more about her ideas and also officially kick off our seminar series.
Thank you so much, Diane. Let me share my screen.
Yeah.
Okay. And then present. What can you see now?
Yes.
Is that okay? Cool.
That's perfect.
Perfect. Thank you so much. I just should look at the camera. This is so weird. Thank you so much, Cheyenne, and to all the ECR organizing committee for organizing this online seminar series and for giving me the opportunity to introduce it. It's so nice seeing it coming together and yes, seeing things starting and people joining. It's something that really I have a passion for, and I would want to kind of work more and bring people together to work more on this cool question. So yes, as Cheyenne said, she asked me to give just a brief introduction on the SPN. She already done it basically, but I will just repeat an introduction apologies for those that had already heard it, but then I will give you an example of my research and when I thought about how a particular aspect of dispersal and mating system might jointly evolve. So for the special topic network, as Shane said, we got the funding from ESEB just over a year ago, and we got started. These are the co-organizers, and some of which are present here today. But now we have quite a few people that have joined all across Europe mainly, and obviously we have here together present the ECR organizing committee that is putting together the seminar series. I probably you all are aware of this, but just to give a very basic introduction, what are dispersal and mating systems and why we are interested in them? So dispersal and mating systems are a suite of physiological, morphological, behavioral, and demographic traits that are quite complex processes made up by a lot of traits and interaction between them. And dispersal has been defined as any individual movement potentially leading to gene flow across space. And mating system, very general, like a general strategy employed in mate choice and acquisition, and which can lead to gene flow across time. And they are both processes that are fundamental for species evolutionary ecology and population viability. So both dispersal and mating system affect a number of common processes that ecological evolutionary processes, and I've already mentioned they both affect gene flow in space and time. And by that, they affect the relatedness structure within an among population, thereby the level of inbreeding, but also the maintenance and distribution across space of genetic variation. And then we can go on and list a number of processes like they affect intraspecific competition, population viability, spatial processes, they affect sexual selection and society evolution, and so on and so forth. I'm sure you can list your favorite process down there. Importantly, these processes are not only affected by dispersal and mating system, but also feedback and drive their evolution. So potentially a lot of common and shared drivers and feedback that could go on between dispersal and mating system. And we know some of them, but actually that space in the middle there is, in my opinion, still a bit empty, because although dispersal and mating system have been huge field of research, apart from some exception, and François will talk to us of one of that exception, they've been quite separated and not so much integrated with each other. And so that brought, again, I want to underline, apart from some exceptions, so it doesn't seem that nothing has been done, but by and large, we have a lack of formal models and syntactic theory that really allow us to understand and make prediction on their joint evolution, on how do they vary in space, spatial processes that affect them, and their equilibrium and feedback. And also we have lack of data that can connect the dots, and especially that can allow us to understand covariation within species. There is typically quite a taxonomic bias and also environmental bias. So, for example, we know very little of dispersal and mating system in marine environment. And this lack of theory and data brings about a lot of knowledge gaps. These are just few examples of how do they covary in space, what will be the impact of the environmental change on dispersal and mating system syndromes, and what will be the consequence for population viability, and so on and so forth. So this is really the background that brought us to apply for the SDN. This is really, I don't know, I probably can use the pointer, yes. This is, oh, it's very small. Anyway, never mind. Now I have a pen. Oops, sorry. Not doing very well here. Okay, no, it's a pen. I'm going to not use the pointer, but you can see this. This illustration is just a schematic, a cartoon, just to illustrate that with the SDN, we want to span the taxonomic divide between plant and animal, this environmental divide, aquatic and terrestrial system, but also process that have been considered. And we try to bring together theoretician and empiricists, those balls there with initial are the co-organizer, but now if we list all of the people that have signed up, we have quite a, we are managing to fill the space quite nicely, although bias still remains. So yes, really, the objective is to foster new synthesis, research avenue, and mainly bringing people together and cross-foster ideas. So hopefully we can address some key questions on dispersal and mating system cooperation. So all I have to say about dispersal and mating system SDN is join us if you are interested. Probably if you're here, you already are. But yeah, this is the QR to our website, and you can just email us and sign up. Okay, so I will switch to my own work. I'm still trying to see if I can go out of this annotate thing and get a pointer, but I don't think, no, okay, never mind. Oh, yeah, sorry, I get it now. Can you see my pointer?
Yes.
Cool, good. Okay, so in my own work, I was really interested, or I'm still really interested, it's not solved at all, the problem on the joint evolution of dispersal and polyandry in the context of inbreeding. And by polyandry, I mean the kind of broad definition of female matings with multiple male in a single reproductive bout. And why dispersal and polyandry? Well, we know that inbreeding is hypothesized to be a driver, especially when there is inbreeding depression, to be a driver of both dispersal and polyandry as means of inbreeding avoidance. And we have theoretical models that show that's possible for dispersal. There are some empirical evidence. And we have some empirical evidence for polyandry, although it's still quite debated. For example, here, example that kind of propelled this hypothesis, like in the field crickets in the early 2000s. And in particular for polyandry, polyandry could lead to inbreeding avoidance in several ways. What I'm thinking here is indirect inbreeding avoidance. This wasn't supposed to come out. Indirect inbreeding avoidance. So it's not inbreeding avoidance by mate choice, where the female decides not to mate with a relative, but by mating with multiple males, the females will reduce the average relatedness among her offspring, and so reduce the risk and the chance opportunity for inbreeding in the future generation. So that's what indirect inbreeding avoidance. However, the only model that was there so far by Cornell and Tregenza concluded that yes, well, this is unlikely because of purging of deleterious mutation that will weaken inbreeding depression. However, that model, well, quite a lot of assumption, as every model, of course, but it wasn't considering spatially structured population, it wasn't considering dispersal. But what interests me is that the share of this potential common driver of inbreeding depression might imply that there might be some feedback between dispersal and polyandry, and when we are in a costas of when inbreeding risk is high. And especially because both dispersal and polyandry are costly, and there are multiple evidence of the cost of dispersal and the cost of polyandry for females, one can hypothesize there could be a negative feedback. So they could be perhaps competing inbreeding avoidance strategy. If you're a very dispersive species, maybe you don't need to pay the cost of being polyandrous, or vice versa. So I wanted to investigate this question, and first I did it with that theoretical model, using a spatially and genetically explicit individual-based model, where I model spatially structured population connected by dispersal. And dispersal and polyandry are evolvable traits, but also inbreeding depression is evolvable, or evolves through accumulation of deleterious recessive mutation. So we end up with a distribution of deleterious mutation in the population. We have the selection coefficient and the dominance coefficient. So you have a lot of mutation that are very, very weak in their effect. But then you have this group of mutation that have a bit of higher selection coefficient, but they're very recessive, including Gretas. And these are the ones that will give inbreeding depression when they become homozygous. So just to jump on the main results, what I found when I ran the model across different costs of dispersal and cost of polyandry is this negative correlation that we hypothesized emerging. So here you have the mean number of female matings, so polyandry, increasing in the x-axis, and the mean dispersal probability on the y-axis. And the colors are different costs of polyandry, so from no cost in purple to high cost in yellow, greeny yellow. And for comparison, you have simulation here in gray, where only dispersal evolves with very low multiple mating, or simulation where only polyandry evolves with very low dispersal. And you can see this negative correlation evolving. So when polyandry is less costly and more polyandry evolves, then dispersal evolves to lower level, and vice versa. When polyandry is really costly and female evolve to mate with few mates, then we have higher dispersal in the system. And this is modulated both by the cost of dispersal and cost of polyandry. So this is what I showed you before, with a cost of dispersal of 0.03. The value per se doesn't matter too much. As you increase the cost of dispersal, here at the bottom, you have, of course, less dispersal evolving, but more polyandry evolving, is still this negative relationship present. Okay, so this was a relatively simple model. Oh, sorry, I forgot this. And of course, the evolving dispersal and polyandry also affect inbreeding depression. So we have this kind of feedback between the three processes. So here on the x-axis, you have neutral homozygosity that can be used as a proxy for the inbreeding level. and probability of reproducing and offspring survival probability, that they were the two fitness traits affected by inbreeding depression in my model. And you can see this group of color lines are different cost of dispersal. So as dispersal is more costly, less dispersal evolved, we have, as we would predict, lower inbreeding depression because there is a lot more inbreeding going on and it is a lot more purging. However, if you look at the different color for each cost of dispersal, different level of polyandry or cost of polyandry and therefore different level of evolving polyandry don't have very much of an effect on inbreeding depression, especially compared to the bigger effect of dispersal. So as I said, it was a relatively simple model, but it showed that in principle, all else being equal, polyandry could evolve in spatially structured populations in direct inbreeding avoidance strategy, and that dispersal and polyandry negatively affect each other evolution in the presence of inbreeding depression. And also the feedback on the level of inbreeding depression, mainly due to the effect of dispersal. So, okay, that was all well and good. And then I thought, okay, but I want to see if it works in an empirical system. So we decided to embark in an experimental evolution project with the seed beetles, Calusbruchus maculatus. And this is all the work of Lana Danan, who was my former PhD student. She defended last year. And she's done an enormous amount of work. As theoretician, I didn't know that the beetles are not the best to do experimental evolution because they have a generation of 1-1. So poor Lana had to work a lot. But anyway, these are some of these. I'm going to present one of the projects she did. So she created this spatially structured population of beetles. So here we have one population would consist of 10 cups, with total of 100 individuals, 10 individuals of equal sex relation in each cup, and they will be connected by tubing that allow this for dispersal. And this is how they look in reality. Not very glamorous, but she built all this up, which I thought was quite impressive. And what we did 3 treatments where we tried to impose different costs of dispersal, high cost, low cost, and no cost, by varying the length and convolution of the tubes. We tested that, and I explain in a second why that leads to different costs. And we also had a comparable, what we call pan-mixed population, so a population of 100 individuals that were fully mixing in a single cup. So what happened at each generation, 5 virgin males and 5 virgin females would be placed in each cup. Then the tubing between the cups will be opened, so they would have 2 hours for to disperse. And at the end of those 2 hours, there is some dispersal mortality, because at the end of 2 hours, the individuals that were left in the tube were eliminated from the system. So quite strong dispersal mortality. Then the tubes will be closed, and individuals in their tub will be left time to mate and oviposit until the adults were dead. When the adults were dead, the infested bean would be transferred in well plates in order to isolate virgin individuals at emergence. They will be left for 3 weeks at 27 degrees, more or less, until, not more or less at 27 degrees, more or less 3 weeks at 27 degrees. And when they emerge, the cycle starts again. We isolate 5 virgin males and 5 virgin females per population, reseeding them in their origin population, and conducted this for 20 generations. Also, every 5 generations, we would do a female remating assay. So from the emergence, virgin females would be placed, each one in a single petri dish with 1 male. The mating interaction will be recorded, then left 24 hours for oviposition, then presented a second male, 24 hours for reposition, and so on and so forth, up to 5 males. Because beyond 5 males, they were stopping mating anyway. And we also do dispersal assay. And in this case, 5 males and 5 females would be placed in the first of a kind of Yes, I'm missing the word now, but an array of tubs connected by very short tubes, and we could measure the dispersal propensity and the dispersal distance in tubs. And also we carry out in block breeding design to measure inbreeding depression in several fitness traits. So the results, turns out that experimental results are much messier than modeling results, which probably any empiricists know. But so what we found in these messy figures that I explain in a second is that female remating propensity and dispersal fluctuating through time in opposite patterns. So here you see the generation on the x-axis and mating propensity on the left y-axis, which are the solid line, and dispersal propensity on the right y-axis, which are the dashed lines. And the colors, as before, so you have blue, green, and purple are low, medium, and high cost of dispersal, while the orange is the permitted population. So you can see here that while mating propensity goes up until generation 15, then goes back again, dispersal fluctuate in the opposite direction, while there's no significant trend in the permitted population. And this is the same more or less for the dispersal distance here on the y-axis. So another thing that you see, which we were very annoyed here, is that you don't see much difference between the three dispersal treatments. So although we saw different costs, and I show that in a second, the dispersal and female mating propensity behave quite with the same dynamic. When we then try to look for this correlation, then we pulled all the treatment, not all the treatment, but all the spatially structured treatment together because they had no difference between them. And we find this negative correlation between dispersal propensity and mating propensity and dispersal distance and mating propensity, which we don't find here in orange in the permitted population. Okay, cool, but what could be driving this relationship? And we have evidence hinting at what could be happening. Of course, our first hypothesis is that there is indirect inbreeding avoidance, there's inbreeding driving this relationship. Well, we have piece of evidence. So we look for inbreeding depression, and there is inbreeding depression in the system, mainly in offspring survival. Here you see inbreeding depression across generation for the different treatment. And offspring survival, we have a lot of inbreeding depression, a little bit in female fecundity, not at all really, we could find in male fertility. And also, although the three treatments behave the same, when we look at the genomic coefficient of inbreeding, we see a differentiation there. We see where we thought we have imposed high cost of dispersal, they are actually more inbred than, well, low cost of dispersal than the stock population. But obviously that's not immediately reflecting on the response between dispersal and polyandry. But another possibility is polyandry evolving as fertility assurance against infertility of males during inbreeding. So what we see is that although we didn't find an inbreeding depression in male fertility, we saw that in the structured population, male fertility decreased through time. And a very nice thing we found is that female allocate eggs homogeneously across mate. So again, this is a very complex figure, but what we see here is for each panel is generation 5, 10, 15, 20. And on the x-axis, there is the first, second, third, and fourth mating of the female. And we measure the number of eggs that laid after each mating. Now, the dashed line indicated the total fecundity across the mating. And so you can see that here all the treatment behave quite similarly at the beginning of the experiment. So the female allocate most of the eggs to the first mate and then quite substantially less to the subsequent one. But as we arrive at generation 20, this pattern has change, especially in the spatially structured population. So the eggs are more widespread across the different matings. And also the fecundity has changed. Obviously, these populations are becoming a small population are becoming quite complex. So yes, we got some evidence in this very laborious experiment that there could be some negative association going on between dispersal and polyandry. It's not as clear as we hope for, and possibly there are multiple mechanisms, some of which we could have evidence, some of others we couldn't properly investigate communication. And so there are a lot of questions open, but also what happens in nature. And if you actually, as a COVID project, we started to pull together literature and look, okay, let's see if we can test it meta-analytically, how dispersal and polyandry may co-vary. And we found that we lacked the data because we found very few studies where dispersal and female multiple mating are measured in the same population. You might have measured for the same species, but will be completely different population. So it becomes very difficult with the current data, or if you know data, please tell me, that'd be super interested to know. But what we found very difficult to test this hypothesis in nature. A few years ago, Jane Reed published a paper that may hint to something like this, but kind of circumstantial evidence. And what she found in a population of song sparrow in Mandarti Island off the west coast of Canada is that recent immigrants to the population, so obviously individuals that had dispersed, had a lower breeding value for multiple paternity. For extra pair paternity. But obviously, there could be a lot of mechanism driving that. So we really need data if we want to test this hypothesis in nature. So I hope I haven't talked for too long. And yeah, so I hope I gave you an example of how there could be complex ecological and evolutionary feedbacks. And we really need to, well, I really like to know more. I think it's important that we investigate. And in that, happy to take questions.
Thank you so much, Greta. So I think we will have some time for the questions, but if we don't finish all the questions right now, we can always ask like after also François' talk. And please just feel free to ask any question.
Will I stop sharing? Sorry, I lost the Zoom. Yeah. Is it still sharing?
Anyone
Yes.
who wants to ask a question could maybe raise their hands and then.
Yes. I'm sorry, I also need to even say the.
Klaus?
Thank you very much. This very interesting talk. I would like to know whether you modeled in the model, whether you modeled the migration of individuals as a trait to be migratory or not, or whether it was a mixed trait where you varied the propensity to migrate. Because in this case, usually it doesn't matter that much for the outcome, but here it really could matter.
So the trait determined the probability that one individual would disperse from its natal site and reproduce. So yeah, it was a probability.
Yeah, thanks.
What I was just about to ask, why do you think it would matter?
The population, or the individuals in the population having some migratory individuals might benefit, because the migratory individuals also bring their genes on another place. And if migration is very costly, maybe the ones that migrate take most of the cost, but the benefit is also to the others. That's a bit my gut feeling why in this case the outcome might differ, because in most of my models, when I
think I can't hear anything anymore.
Yeah, I think you muted yourself. Oh no, but we don't hear you anymore somehow.
Sorry, Klaus, we can't hear you.
Now?
Oh,
Yes.
now yes.
Okay.
Yeah, we lost a bit the last
yeah.
bit.
I think that the cost is mainly to the migratory individuals, but the other individuals of the population that are related to the migratory individuals also benefit from the migratory individuals. And that's why I think it might be a lower frequency if it's not a mixed trait, not a propensity, but there's some individuals that migrate, a genotype that makes them to migrate and others not. Then I think my gut feeling is that the proportion of individuals migrating might be lower. That's why I ask.
Okay, I see.
Not sure,
So,
but it's just
so
my
there
gut
might be
feeling.
two different strategies going on in the population at that point.
Yeah, where there's two different strategies, or all the individuals have a propensity, a certain proportion. And this could be one of the few examples I know where it really matters. And that's why I wanted to know whether you looked at it. Thanks.
Yeah, no, I didn't. Thank you.
Is there? Okay, Shang-Yi.
Hello, thanks, Greta, for this very interesting talk. I have a question, really naive, related to the experimental evolution experiments. When you connected this local habitats with the tubes and you let the individual beetles disperse for 2 hours, and then you remove the tube and individuals still inside the tube. In this case, I'm wondering if you are actually selecting for individuals that either dispersed very quickly or stayed. So
yes, that there is a possibility that there is. I mean, yeah, there is a possibility that.
I was just wondering if if you're selecting two very different populations at the same time, then that might contribute to that you don't see much patterns eventually.
Yeah, it would still be a selection in individual that disperse, right? Or at least on a proportion of individual that disperse.
In that case, you would, for example, only allow those individuals that disperse to reproduce, but right? If they haven't dispersed, they can still stay in the local habitat and still reproduce. This is just, yeah.
Yeah, but yeah.
Yeah, thank you.
Thank you.
So we have another question from June.
Yeah, thanks very much, Greta, for an interesting talk. I just had a question, maybe I missed it, but to what extent do you know the heritability of dispersal, and how easy would it actually be to estimate that? Maybe it's not so difficult to actually take individuals with different propensities to disperse, those that don't disperse versus those that do disperse, for example, and with a simple crossing design, have a look at the dispersibility progeny. Have you done that?
A simple crossing design. It sounded simple in principle, but we haven't gone. We did it, but we haven't gone much far, and the heritability was very low, actually.
Yeah.
So there is also a possibility. This is a lab population that has been in the lab for decades, right? So there is a possibility that the variance was too low to really see changes. And I didn't mention that there is also the possibility that these responses are not just evolutionary, right? It could be some plastic responses. But yeah, we failed a bit with the heritability.
Yeah, because I think obviously the heritability is absolutely critical in an experiment like this. I mean, I don't know how the plasticity. Are you suggesting that there was some sort of maternally transmitted plastic response? Is that what you're suggesting?
Yeah, that's what we can't exclude, right?
Mm-hmm.
Yeah.
Yeah. Yeah, I mean, it's interesting. I mean, it would be interesting because if you don't find the responses you're predicting, but you find that actually there's very little heritability, then that immediately provides a simple explanation, even if there's strong selection for
Yes.
the evolution of dispersal. If there's no variation for that trait, then obviously nothing's going to evolve in a few generations like that. So
yeah, so if I remember correctly, we found some. It wasn't zero. We found some heritability for dispersal. It was quite complicated. Now I don't remember exactly the ins and outs. But yeah, obviously we are not on the high side.
Yeah, we've been doing experimental evolution on sex allocation in a plant species, and there's been a very strong response to selection, but the heritabilities are 40%, very high actually.
Oh, wow.
Okay. So that makes it much easier to detect. I mean, the thing about plants, and I mean, you're actually, with 1 month per generation, you're well ahead of what we can achieve with our plants. But we have very high heritabilities, and if you've got small heritabilities, that limits what you're going to see in a few generations.
What I've been wondering also is how strong is the selective pressure of this treatment, right? We are not strongly selected from one trait. We are creating a spatially structured environment.
Mm-hmm.
So there are a lot of things that can be selected for. So that's what I'm also wondering, whether, it goes a bit out in the wash of how different traits that we are not aware that we could be selecting. We were not doing kind of artificial selection, right? We were not actively selecting for the dispersive one. So yeah, I thought it's still interesting that even with all this problem, we still see this negative correlation between the system that evolved more female multiple mating and system that evolved more dispersal.
Mm-hmm. Yeah, absolutely. Thanks very much.
Yeah, the lack of difference between the treatment after all the working out how we could impose cost of dispersal was a bit disappointing.
Yeah.
There's one question in the chat. Marta asks, what data would you need to be available for the same population?
Yes, so we would need some estimate of dispersal, propensity. Probably, I guess, would be the easier. I guess depends on the study system, and in the same population, some measure of usually is multiple paternity, right? Usually you don't have data on the matings, but on the multiple paternity, basically.
So I think we would move to the sorry. Shall we just have one more last question, then we move to the second talk, just in case? Yeah. So Jana.
Okay, thank you for the really interesting talk. So maybe I missed it, in which case apologies. But if I understand correctly, both in the simulations and in the empirical thing, you mostly looked at like a correlation between like simulation runs or like populations that evolved mating, polyandry versus dispersal, right? I was wondering what happens like at the individual level. Do you see that like individuals who disperse more then go for monandry and vice versa? Like, would you expect that to be a negative correlation also within populations, or yeah?
Super good question. Yes. Yeah, it would have been nice. No. So both in the models and in the experiment, well, I guess in the experiment we couldn't test correlation individual level, but at least in the model, I didn't find any correlation at the individual level and any correlation of within population. The only correlation was among replicates. So either a system was evolving higher dispersal, less polyandry, or vice versa. I mean, no longer continuum, right? But I couldn't find any spatial structure. I mean, the environment was very simple, right? It was homogeneous environment, was suitable in the model, and any individual relationship.
Okay, thank you.
Thank you.
So thank you again for Greta. And if you have any question to Greta, just feel free to maybe type in the chat. So we're also gonna have another round of maybe discussion after François' talk. So now let's just move to the second one. And so I would like to introduce our second speaker, Dr. François Massol. I hope I pronounced it correctly. And so he completed his PhD at the University of Montpellier, and then he pursued his postdoc at the University of Texas and Austin. And he's currently a researcher at the University of Lille. And his research mainly focuses on evolutionary ecology, particularly in species interaction networks, spatially structured ecological systems, and the diversity of the stability of the systems. And today he will share us about his perspective on dispersal and mating systems. Now, so François, just feel free to start.
Thank you. Thank you very much for the invitation and the introduction. Yeah, I hope you all hear me fine. So today I'm gonna show you some results we obtained with mainly with Pierre-Olivier Cheptou, with whom I worked for quite some time on modeling the joint evolution of selfing and dispersal under uncertain pollination conditions. And before I start, there's a big disclaimer, which is everything I'm going to show is about models we developed like around, I mean, a little bit more than 15 years ago. And I haven't been doing a lot of things since then on that. So, I mean, it's models that actually that we did that were fine at the time, and just so that you know that it's not like completely brand new. And also it's going to be a lot of modeling, and especially like a modeling that is explored through mathematical analysis. So yeah, I try not to scare anybody with equations, but just that was the disclaimer. And so the idea of the model is to look at self-fertilization, so what we also call selfing, and dispersal. And dispersal and selfing are two major traits. So traits that vary within and between species. So I mean, most of the illustrations will be about plants because well, we had this bias with Pierre-Olivier to actually think plants and then pollination. But in plants, you see like papers on some seeds and seeds that don't have them that actually disperse at a different distance. And also you know probably that some species of plants have different selfing rates. Some of them never self, so are complete outcrossers. Some of them always self-fertilize, some do something in between. And so the modifications of these traits are expected to greatly affect the fitness of the organism and their evolution. So the evolution of these two traits are often considered separately, as Greta said, in theoretical models. So it was kind of the challenges was to actually understand that. So just to clear things completely, like these are the classic factors that are known to affect the evolution of dispersal. So the list is not completely exhaustive, but mainly it's the basic things. So kin competition, habitat heterogeneity, fluctuating environment, costly dispersal, the chance of inbreeding, and the abundance of local catastrophes and population dynamics. So if you put everything together, yeah, some kind of like, and trying to balance things, you have on one hand, like on the pro, so all the selective pressure acting for more dispersal are mostly avoiding inbreeding because of kin competition. So you try to evade competing with your kin. And then there is everything that has to do with variability in time of the environment. The environment in a broad sense, meaning both population or community dynamics that change in time, and also the fact that the abiotic environment could change, so which is subsumed in the environmental viability or catastrophes box. And on the other side, so for the things that act against dispersal, you get environmental heterogeneity, which means in space, not in time, and the cost of dispersal. And for selfing, we can do it, but it's much simpler. In fact, you have like 3 main lines of argument for or against selfing. You have actually 2 for selfing, which is a higher transmission rate for the genes, and for plants in particular, you have this the fact that you have uncertainty in conditions for pollination if you rely on pollinators or on wind for pollination, and on the other hand, you have inbreeding depression acting against the evolution of self-fertilization. So basically, the balance looks like something like this. So with 2 arguments for more selfing, 1 against it. And so one of the things that triggered this whole work was that there is this law called Baker's law, which well, basically it articulates the fact that selfing provides reproductive assurance without pollination. And so the idea is that you expect island species to be selfers. And also, so this is a corollary belief of that, would be that selfers should disperse more. And that was kind of the ignition of the work we did with Pierre-Olivier, was to actually look whether this can be modeled and actually whether we could get back this type of results, especially since when we looked at the literature, you get mixed evidence for that. Pollen limitation is actually very common in plants, and you have a high proportion of self-compatible and a low proportion of self-incompatible species on islands, which looks like it could be according to Baker's law, but also you have predominantly selfing species which are slightly associated with colonizing ability. And also you have a very high incidence of dioecy on islands. And so for all of these reasons, it was not very clear whether actually Baker's law is true in nature, or whether actually the model that is behind that, so the implicit verbal model, actually works. So we were interested in actually formalizing it in a very simple way to look at the predictions of such a model. And also just to wrap up, when you have the joint evolution of 2 traits, so in our case here it's selfing and dispersal, these traits could evolve for a multiplicity of reasons. So reciprocal selective pressures, shared selective pressures. So reciprocal meaning the one of the traits acts on the selective pressures of the other, and shared selective pressures meaning you have one selective pressure that actually acts on both at once. But you could also have allocation trade-offs, the genetic correlations, structural constraints like allometry that actually affect the fact that you have links between traits. But for today, so for the model that I'm going to present, there is no trade-off or genetic correlation or structural constraints. It's mostly about selective pressures. So we are looking at a model where we want to understand what the selective pressures are doing to the traits. And in our case, so for plants, and we want to look at selfing and dispersal, well, the most obvious selective pressure that you want to look at is pollinator dynamics or pollinator paucity, because you have, on the one hand, if you look at dispersal, if you consider a species that is mostly outcrossing and that actually needs the pollinators, every time you don't have the pollinators, it means it's a catastrophe, actually, because you can't really reproduce. And we know from models about the evolution of dispersal that the more catastrophes you bring about, the higher the selective pressures to disperse. On the other hand, if you look at selfing rates, if pollination is uncertain, it's kind of a boost to actually improve your selfing rate. So it's a selective pressure for selfing, because with selfing you can maybe you do worse because of inbreeding depression, but you will never do worse than not reproducing at all. Okay, which could happen if you rely too much on pollination. And the thing is that you also expect reciprocal selective pressures in this case, because if your selfing rate increases, it also means then you don't really see this variability in pollination. You don't see it anymore. And so maybe you should not disperse as much. And reciprocally, if you disperse a lot, then you don't really feel that pollination uncertainty, because you will hedge your bets and have lots of your offspring in different conditions where pollination could be different, and actually some of them could be pollinated, and maybe then you don't really need to self. Okay, so that's the thing about this pollinator dynamics, is that they are both a common selective pressure and also they induce reciprocal selective pressures. And so the whole question is about the evolution of dispersal and selfing rates, whether these are intertwined under pollination uncertainty and how. And so, as I said, it's mostly based on two papers we published. So the first one in 2009 and the second one in 2011. And so these models, if I am to really sum up the ingredients, they are dealing with an infinite island model with large populations. And basically, we have 4 different parameters. The first one is E, it's pollinator paucity. So it's how often you are in a situation where you don't have pollinators, so you can't really outcross. Another parameter is delta, so it's inbreeding depression. So we did it really in the simplest way. So inbreeding depression never changes in this model. It's just assumed it's a parameter. You have a parameter called phi. So it's the autocorrelation of the pollinator paucity condition. So I get back to that in a minute, but basically it's to measure the dynamics. So to parameterize the dynamics of the streaks with or without pollinators. And you have a last parameter, which is migrant or disperser survival, which is named Q. And the 2 traits under selection are S for selfing and D for dispersal. So very simple. And so we are measuring variability and heterogeneity with these 2 parameters. So E is measuring like the fraction of patches that don't have pollinators at any time, but these changing all the time. So some of the patches could get pollinated, while others could get a catastrophe and not have pollinators anymore. And the second measure is the phi. So the phi parameter is there to see whether this variability is completely random. So it means phi equals 0. So it's the first time series you see at the top. And if phi is actually positive, so it means you have positive autocorrelation. It means that when you're on a streak with pollinators, it's gonna last. But when you're on a streak when you don't have pollinators, it's also gonna last. And when you have negative phi, so the last one at the bottom, it means it's gonna alternate much more than expected. So actually, you're gonna have without pollinator conditions in the same patch, alternating much more regularly than expected. And so we've followed through with mathematical analysis of the model using the adaptive dynamics classic assumptions. So basically, we're using what is called the phenotypic gambit. So we're using the model as if we don't really care about genetic architecture or the fact that you have links between locus or whatever. So it's really the simplest way of looking at that, with rare mutations, and every mutation has very small effects. And to actually solve that, you need an expression for the fitness, and I get to that a little bit later. And once you have the fitness, you get the selection gradient that helps you ascertain which strategies are singular. And then you can look at the convergence stability. So whether you get to these particular strategies or in the first place. And then you look at the second derivative of the fitness, so called the Hessian of the mutant fitness. And then it gives you an answer to the question whether these strategies are ESS, so evolutionary stable strategies, or not. And so to get the fitness, so the idea is that because it's a cycle of events one after the other, you have to actually compute some kind of matrix for the proportion, for the abundance of individuals or of patches occupied in the different states and that actually reproduce. And so to get to that, so you compute your matrix based on the events. So here we did it with reproduction first, then dispersal, then regulation, and then change. And so possible change, it actually doesn't have to happen all the time. And once you have the matrix, you look at the dominant eigenvalue, and it gives you the fitness. And to get the matrix, you need to do the product of different matrices, one for each of the events. So it's actually not so complicated, like set that on paper. The computation is actually sometimes a bit more tricky, but I'll not get into the details of that. And so for the results, what we found basically in the simplest variation of the model is that we have 3 scenarios that could occur. So in this type of plot, so here I plot the actual selection gradient. So all the arrows are selection gradients. And you see on the x-axis is selfing rate, on the y-axis is dispersal rate. And the first scenario is that you evolve towards no selfing and some dispersal. And the good thing here is that we can actually compute how much dispersal you will get. It's a simple formula that you can get from the model. And so this is the first scenario. The second one is the total opposite. So it means you have some selfing, you have no dispersal at all. And also in this case, we can get the ESS for the selfing rate. And sometimes we don't have like 100% selfing. So we might have mixed selfing with this model. And you have the first scenario, which is it's the tricky one, where you actually have the 2 possibilities. So being disperser and outcrosser, or being partially selfer and not dispersing at all. And in the middle, you have something called an evolutionary repeller. So a strategy that really repel everything. And the thing is, in this case, you have alternative stable states. So you can go either to the blue one or the red one among the 2 strategies. And it depends on where you started from. And the thing is that you have a strong pressure against selfing and dispersal because you don't want to handle both the cost of selfing and the cost of dispersal. And you have a pressure for either selfing or dispersing. And depending on what you do, you become some kind of either a generalist of the pollination problem, if you want. When you are a selfer, actually, you don't see the difference between the patch that have or don't have the pollination. So you're kind of a generalist of the environmental conditions. And when you go to the dispersing strategy, actually you spread your spring across a very heterogeneous environment. And so this also means that you don't really, you don't see the uncertainty in pollination condition just because you hedge your bets. And as I said, the where you end up depends on the initial condition. And so it's called, it's also called alternative stable evolutionary equilibria. So if we were to actually represent that in the parameter space of, so here the x-axis is pollinator paucity. So if you go to the left, it means you have pollinators all the time. To the right, you never have them. And migrant survival, so on the y-axis, So at the top, they all survive all the time. At the bottom, they never survive. Then you see you have the three strategies. So 1, 2, 3. So 1 is dispersors, 2 is selfers, and 3 is the alternative one. You can, if you follow the horizontal dashed lines, it goes to what you have on the right-hand panel. So actually, the value for dispersal and the value for selfing. So one thing you see is that you really never have any selfing when you are on the disperser outcrosser strategy. It's always 0% selfing. And for the 0% dispersal, you can have mixed, partial selfing, and then at some point it goes to 100% selfing. And one thing that you also see when you look at a plot like that is that there is an evolutionary priority effect or hysteresis, meaning that if you change the environmental conditions, so here say you increase the probability of being pollinated, so going from right to left, it's not you're gonna stay longer with the selfing strategy. So the ones where you actually never disperse. But once you get to the outcrosser strategy, and then you decrease the probability of being pollinated, you will stay on this branch. So stay with the disperser outcrosser strategy longer when you go right. And so it's called hysteresis. So it's the fact that you don't get exactly the same thing depending on where you started. When you add autocorrelation to the picture, actually so here it's pollinator paucity on the x-axis and the autocorrelation in pollination condition on the y-axis. So the two regions in complete white here are impossible. So the ones on the bottom left and bottom right corners are impossible. You can't really have that. But you see you have the three different scenarios that are still there. The gray level is for the value of dispersal. So the whiter, the less dispersal you get. And so basically the conclusions are the same. So you still have the three scenarios. You have the two syndromes, so dispersal outcrossing versus no dispersal selfing. And the thing is that the autocorrelation parameters mostly only affects the value of dispersal, but not of selfing. And one thing it does is that under the intermediate scenario, so scenario 3, it's the priority effect is even more drastic when you have autocorrelation, in the sense that it's kind of all or nothing, okay? And so basically the take-home messages of these models are we have three scenarios for evolutionary equilibria. So it's kind of, so it's out of models that make a lot of assumptions. So you have to take that with a grain of salt, but basically that's what we found. We have two syndromes for trait association, so dispersal outcrossing and no dispersal partial selfing. And all of that goes against Baker's law in a sense. And under the third scenario, so we have bistability and hysteresis. And negative autocorrelation selects for more dispersal, and positive autocorrelation disfavors the outcrossers. So that's what we found. So after that, I kind of read what happened after we made these models, and there were actually at least two models that were published after ours. So with Ryosuke Iritani, Pierre-Olivier published a paper where they did the same thing, but with environment-dependent inbreeding depression. And they found results that are a bit different from ours, meaning that you can get back things that are more in accordance with Baker's law if you allow the inbreeding depression to depend on the environment. And Roger et al. Did another variation of the model where they introduced the density of individuals within patch, to look at the allele effects and the effect of competition, density-dependent competition within the patches. And with that, they found a positive relationship between selfing and dispersal. So when you introduce these components in the model. And lastly, one thing that we did with Pierre-Olivier and Maxime Dubar, who was doing an internship with me, and at some point, we looked at the same model, but introduced kin competition. So actually having a number of individuals per patch. And the thing, so here in the two plots, you have on the x-axis, you have the patch size, so the number of individuals per patch. And the y-axis is the ESS for the dispersal rate. And so as before, you also have these two syndromes. So either you outcross or you partially self. And the thing is that sometimes, and especially at high autocorrelation, so it's on the right-hand side, so the right-hand panel, you see that sometimes actually the selfers disperse more than the outcrosser in this model. And so actually with kin competition, what we introduce is a general pressure to disperse, because with kin competition, everybody has to disperse at least a little bit, because you want to avoid to get your offspring in competition with one another. And the thing is that with selfers, you have twice the relatedness of the ones of outcrossers. So there's an intrinsic higher selective pressure due to kin competition to disperse. The thing is that this pressure is actually very low when you are at high number of individuals per patch. So that's why we didn't see it before. But when you get down to like between 1 or 5 individuals per patch, it can be seen actually. And so it's a balance between kin competition and pollination uncertainty. And it depends a little bit on autocorrelation for reasons I won't have time to elaborate now. So the perspective of this kind of work is that we need to actually separate the extrinsic and intrinsic determinants of pollen limitation. So that's one of the things that the paper of Roger et al. Began to do. But I guess we, in terms of density, we have to use this to actually have an effect on competition, on allele effect, but also prove relatedness on the kin competition, as I just showed you. And the other thing is that in animal pollinated plants, we need to account for the networks of plants and pollinators, and maybe you would have like pollinators that are more or less reliable than others, and this could change a little bit the type of model to use. And also the evolutionary feedbacks between like other traits and the ones I've just shown you, so selfing and dispersal. So for example, the evolution of visitation rates in the pollinators could go, could be selected by certain characteristics in the plants, and that reciprocally could affect the evolution of selfing and dispersal. And at that, I will, thank you for your attention. I'm happy to take questions.
Thank you so much, François, for your really nice talk, and especially the future perspective. I think that's also the major goal we want to have for our seminar. And so now we will move to anyone have like questions for François' talk. So John.
Yeah, thanks very much, François. Very nice to see your talk and remind me of these models that you did. I guess my questions are more comments. I think what we maybe are seeing from the different models that are coming out is just how complex these scenarios actually are. And for example, one needs to make models that are simple enough to make sense of, but the risk is missing out on variables that actually probably make a lot of that are really important in the wild. And the one variable, for example, that immediately jumps out for some of these models, and I've made models that make this assumption as well, and that is fixed inbreeding depression. And I've begun to think that actually any model of a subdivided population that tries to get at an ESS and assumes fixed inbreeding depression is going to yield results that may be not very relevant to nature, because we know that inbreeding depression evolves with the mating system. And so if the mating system is evolving towards selfing, we expect that the inbreeding depression will evolve towards lower values and so on. So, so that'd be my first comment, that we, looking to the future, I think what we really need to be doing is being very careful about some of these assumptions about what is dynamic in terms of inbreeding depression and what isn't. And maybe just a further comment about that, because I think this is particularly important for inbreeding depression. In a subdivided population model like the ones that are important for dispersal, of course, you can have populations that are established by inbred individuals. And so you end up with a population that may actually have almost no diversity whatsoever. There may be high genetic load expressed by those individuals, but technically there's no inbreeding depression because any outcrossing is equivalent to selfing in a population like that. And that, I think, I can't see how one could model that without being absolutely explicit about the genetics of inbreeding depression. And so the phenotypic gambit approach seems to me just not adequate when we're dealing with inbreeding depression in models. And maybe you can comment on that, because you know a lot more about modeling based on these phenotypic approaches.
Yeah, thank you. Yeah. I mean, for sure, it's quite simplistic in the way we look at inbreeding depression, and of course, yeah, we're missing something. So regarding the fact that inbreeding depression is going to change in time and also in space, it's for a variety of reasons. But the thing is also the fact that inbreeding depression is really, I mean, the definition of inbreeding depression is very pragmatic in a way, because it's like, okay, what's the fitness of outcrossers versus the ones of selfer, and the difference, the relative difference tells you what you miss. So it's something that actually pertains to what you can actually measure in the field. The thing is, in a model, maybe you want to be more like absolute, in the sense that you want to actually model the what you lose on like generally from being a selfer in a particular species, which has a genetic background that has a certain variety of deleterious mutations that are like scattered in the genome, and that's gonna crop up if you self too much. So that's one thing, but that's not actually answering everything. The other part of your question being populations funded by inbred, it's sure. I mean, you don't see the inbreeding depression as long as you have only inbred or so selfers actually in the populations. But if the population then receives migrants that are not completely inbred and selfers, then you immediately realize the inbreeding depression. So the thing is, maybe there is this happy phase, I don't know how long, that actually change, and that would be that makes a difference for the selfers. That's one of the things that maybe that's needed to actually change in the model, is that to actually model what happens during this phase and whether it's important for the dynamics of selfing. Because it could be that like this phase is long enough that selfers have this early start advantage all the time, and that is sufficient for selfing to be actually a very good strategy. We haven't put that in the model, so we can't really argue for or against. But sure, it's gonna probably be very needed to actually look at things in nature, and in particular in plants on islands, it's something that we didn't want to put in the model at first because we wanted something very simple, but through it's much it's kind of natural to add that at some point.
Yeah, thanks. Maybe also, yeah, I totally agree with you. And what we really need are these, more complex models. But the problem is, as soon as you include that complexity, they become more difficult to understand. The other comment I wanted to make was just in terms of Baker's law, and I guess this is just a perspective thing. But I think my reading of Baker's paper is that it's not so much the rate of selfing, and I don't think he ever referred to the rate of selfing in his two papers on Baker's law, but rather the capacity for selfing. And I think that, for example, the increased frequency of diece on islands, that there are lots of reasons that might account for that, but one thing to bear in mind is that sometimes dioecious plants are generally outcrossing, but they may have a capacity for selfing if they are inconstant in their sex expression. And I think also with species that are self-compatible but outcrosses, a small amount of selfing might be sufficient to allow colonization of islands. And I think that was always my sense with Baker's law, that he was really focusing on an ability of organisms to self-fertilize rather than locking themselves into a strategy of selfing with a high rate. And so I don't think necessarily the predictions from models that point to an association between outcrossing and colonization, or outcrossing and dispersal, necessarily count against Baker's law. I think Baker's law is not as strong a claim. I don't think it's making such a strong claim as sometimes is stated.
Yeah, I mean, you're completely right. It's just, it was to actually make the history of the papers and why we started these things. But after I mean, for this kind of opinion synthesis we did in 2011 was we actually reread Baker's law, and there's what people read in it, and there's what actually written. And the thing is, yes, it's not completely clear whether you expect anything in terms of syndrome from Baker's law. But the fact, that a little bit of selfing can help you, for sure, it's somewhere there. The thing is that if you only keep like this ability for selfing, but almost never use it, in terms of inbreeding depression, is gonna keep. I mean, it's not gonna help you purge. And so the inbreeding depression, if you had one to begin with, it's gonna stay very high. So there's this thing, but I totally agree. Like Baker's has been read differently by different people.
Right. Anyway, thanks for a very interesting summary of these papers. It was a very nice seminar.
Thanks.
Thank you so much for the interesting discussion. So this question is from Luis.
Yeah. Also, thank you very much for the presentation. I found it super interesting, especially because it's a type of model I'm not super used to. And one of them being that it seems like in this model, for instance, for the dispersal rate or the selfing rate at equilibrium, it really depends on some parameters related to this pollination. And I was just wondering, this pollination uncertainty, it seems like in plants, some have really specialized pollinators and some are very much generalist in terms of pollinators. And so in which case, I wonder how important would it be to consider this pollination uncertainty? And I was wondering whether you had any thoughts on that. Because,
Okay, so it's really a matter of interpretation, I guess, of the parameters. But if you think like the parameter for pollination, so pollination paucity is about like when do you not get any pollinator at all? And then you understand you have like different pollinators, and some of them could be there or not. Then for sure, if you're a generalist plant that has a lot of different pollinators, you're probably less affected by this problem because you're attracting different pollinators, and so you're probably less likely to suffer this. So less likely to suffer the problem of pollination uncertainty. The thing is, it's not totally clear whether when you are a pollination generalist, so a plant that actually attracts a lot of different pollinators, whether these pollinators in turn are also generalists among plant species or not. So for example, I was involved at some point in a data analysis on European orchids. And what we saw is that the orchids that are generalist in terms of pollinators have pollinators that sometimes are very specialized. And on the contrary, the pollinators that are specialized on the plants that are specialized on one pollinator, they use a pollinator that actually is a very good generalist. I mean, it goes to different kinds of plants and not necessarily orchids. So in this case, it was not positively correlated. And then you can say, okay, well, yes, the pollinator is probably going to be more reliable if it's a generalist, because it will find plants and so things to feed on in different plant species. And so maybe you don't have this problem of not finding it. And conversely, if you rely on a lot of specialist pollinators, well, at least one of them is going to be there. So it's two strategies to try to stay on top of things and not have this uncertainty. But what we don't put in the picture when you look at that is, okay, how much cross-pollinating, I mean, between species, so things that actually amount to nothing, is going to happen. Because if you have a generalist pollinator, then your generalist pollinator is going to put pollen from different species on the plants, and that's going to lead to nothing. So that's one of the things. I mean, we didn't put that in the model, obviously, but that's one of the questions we at some point we were asking with colleagues in Lille, that how the competition for attractiveness and for pollinators between plants can actually has an effect on the selective pressures for or against selfing in this kind of context. You can actually control more or less the uncertainty depending on the types of pollinators you select as your preferred pollinators. But then there is this feedback on selfing. So yeah, I mean it's a tough question.
It's super interesting.
Thank you.
Yeah.
So Xiangyi.
Thanks, François, for this very interesting talk. I found it very interesting and a little bit puzzled by one of the equilibrium condition you found, that is no dispersal and some selfing, but not completely. Because you have this infinite island model, my intuition is that because of king competition, you kind of like always can benefit from some dispersal, and why it evolves to completely no dispersal. And another thing is, if nobody is dispersing, why would you not completely do selfing? But like this scenario 2 in your case is interesting for me.
Yeah, actually the scenario 2, the first results I showed you is for very large populations, where you actually, I mean, you don't feel the kin competition, so you don't disperse. But then when at the very end, I showed you what happens when you introduce kin competition by having a limited amount of individuals per patch, and then yes, everybody disperses. And then the kind of the additional question is, okay, in some particular conditions, is it going to be the selfer strategy that disperse more, or the the outcrosser one. And what we found is that actually it depends on the autocorrelation parameter, because in the like you have this difference between the like if the autocorrelation is very negative, then for the outcrosser, I mean, it's always a boon to disperse, so they're gonna disperse much more than the selfers. But then if the autocorrelation is positive, then if the outcrosser lands in a patch that is like temporarily pollinated, then it has less incentive than the selfer to disperse, because the selfers have like twice the relatedness, and so more kin competition effect.
Yes.
So that was one of the things we added in the very last things. But it's as I said, it's not yet published, but it's one of the tricky things that we added at the end.
Thanks a lot.
Thank you.
Do we have any more questions for either François or Greta? So if we don't have any question, so actually it's also already like around 4:30. And I really thanks again for everyone. Like this is the first seminar we have, and it's so great actually to have so many of you here. And another thing, I think I would like to, maybe it's a bit too late, but I still want to introduce like all of like our organizers for this seminar to happen. Dania, Ella, who are based in University of Bern and who are PhD of Xiangyi. And we have also Luis, who also one of our organizers. And so to mention that, because we are also really keen to know if you have any interesting topic, especially about mating evolution of co-evolution of dispersal and the mating system, just feel free to reach out any of us. And if you have any recommended speaker which you really wanna know about their work, just feel free to let us know. And for the next one, like we will gonna have our second seminar in around like April. The topics will be about sexual selection and sex-biased dispersal. And just we will keep you updated, and just follow our website or follow our updates. And then just you are really, really come again, and we are so happy to see you again. And I don't know if anyone else has anything to say before we finish.
Thank you, Cheyenne.
No problem.
Thank you, François, as well.
Thank you so much, everyone.
Thank you.
Bye-bye.
See you again. Have a nice day.
Bye.
Ciao, ciao.